So, welcome everyone to this EAAP webinar focused on the contribution and application of the Zebrafish model to the One Health concept. So I am Maria Isabelle Toulouse, Deputy Coordinator of VetBioNet, and it's a pleasure to present and share this webinar with Christelle Langevin from INRAE. So this webinar is a little bit special for several reasons. And the first one is that it's the last webinar of the 2021 webinar series hosted by EAAP each month. And it suggests an exciting new agenda for 2022. So the second reason is that it's a joint meeting with VetBioNet, which is an European community funded infrastructure network, and EAAP is one of our partners. And so I will take a few minutes to introduce VetBioNet, but before I would like to thank Andrea, Ricardo, and Federico for offering to join EAAP for this event. And in particular because we have chosen to address a somewhat different topic than the usual EAAP meeting sessions by focusing on the Zebrafish model. So in short, so VetBioNet is a consortium, VetBioNet for Veterinary Biocontained Research Facility Network is a network infrastructure that is funded by the Europe, the H2020 program. So it's with the overall goal is to establish a comprehensive network of European research infrastructure sites, international organization and industry partners that is dedicated to advanced research on epizootic and zoonotic diseases, and also that promote technological development. So VetBioNet gather a total of 30 academic and industrial partners from EU member states, but also associated and third countries, and also non-European countries. And as you can see here, there are many institutions that are expert and who play a major and well-known role in the field of infectious diseases. So this network has different specific aims, such as preparedness of to re-emerging epizootic and zoonotic disease outbreaks, harmonization of best practice and the use and promotion of the use of global standards, ethical standards, and many things like that. But I would like to highlight the fact that one of the main objectives of our network is to promote and facilitate the access to transnational access to the infrastructure resources of the network. And this includes BSL-free animal facilities and laboratories, technological platforms, and sample collections. So, and what is interesting what could be interesting for many of you is that it provides access, a free of charge access to our facilities. So 23 installation, 55 access providers. There are BSL-free facilities, but not only, we also propose access to analytical platforms. And the only requirement is to propose a sound project with epizootic and zoonotic disease, and this includes African swine fever, influenza, Welsh night fever, and fish disease, and other animals diseases. So there is the offer is the portfolio is quite large, so it can be the use of cells from different animals, but also larger animals. And the wide range, the animal panel is quite large because there are classical animal models such as ferret and rodents, but you can also give access to livestock and wild species, as you can see here. So there is currently a call. This is a final call, and you can apply by going to directly the link in the VetBioNet website. And please feel free to apply. It's quite easy way to apply. The deadline is close, is very soon, but there might be a further extension depending on the nature and the eligibility of the projects that we received. So coming back I just wanted to also to highlight the fact that VetBioNet will take part of a larger consortium of networks that has been recently accepted by the European community and will be found by the European. And the kickoff will be in February of March. And this will be the opportunity to have access to a very large portfolio of research infrastructure services. And again, with very very large catalog of infrastructure, and this will be mainly open to emerging disease, because the objective is to propose integrated services for infections disease outbreak research. So please go to the Easy Door documentation that will be online very soon. So coming back to our webinar today, so this is, I have to say also that it is the first of a series of 3 webinars that will be organized by VetBioNet. And this series will focus on experimental models that for studying infections develops alternatives to animal testing. And because this is an important focus of VetBioNet related to the 3 R's goals. So as you know, today we'll speak about Zebrafish. And Zebrafish is a model that has been widely used to study neuronal and immune system development. But today what we'll see and discuss is how the use of the Zebrafish larvae is a surprisingly relevant model for understanding aspects of host-microorganism interaction. And this is quite interesting because zebrafish larvae are not considered as animals per se, and this is a very so that's why zebrafish larvae is emerging as an innovative and attractive model, and very well used in the context of the 3R. And so this is used both for basic research studies, but also for the development of screen for drugs and other components. So today we will focus on zebrafish, and Christelle will present the in vivo model for biomedical and animal research, health research in general. And then we will focus on host microbiota interaction, thanks to David Pérez Pascual, to finally have a general overview from pathogenesis to cell biology, using zebrafish as a model of infections. So some organizational issues. So as you can see here, the webinar will be recorded and will be available on the EAAP website for members. And we'll see also for the VetBioNet website whether it will be possible to post it. We plan after each presentation 10-minute questions and answer, and please raise your questions in the box, and preferentially the Q&A box, please. Please turn off your video and microphone during the event. And so I take also the opportunity to highlight the next EAAP webinars that will take place in the 11th of January next year. And also ask you to join our next webinar. Will take place in March 2022, and we will define more specifically the date later on. So before giving the floor to Christelle, I would like to thank Christelle, Vincenzo, and David for having accepted to illustrate the different facets of the use of zebrafish larvae by presenting their work, and they were very enthusiastic, and we are very glad to hear you. So first, Christelle Langevin will present her work on zebrafish, and she will present how zebrafish is an interesting in vivo model for biomedical and animal health research. So briefly, Christelle is the scientific manager of the in vivo and clearing imaging platform, which belongs to INRAE and the National Research Infrastructure Emerging. For 10 years, she develops zebrafish models for in vivo investigation of infectious and inflammatory pathology, advanced 3D imaging methods, and she uses also zebrafish for the screening of drugs targeting human and animal diseases. Christelle Langevin is involved in national and international projects, and she is a member of national bioimaging networks. So, Christelle, the floor is yours.
So I try to share my screen. So you just tell me if it's working or not. Is it working?
It's not in presentation mode.
Okay,
yeah.
So I have to stop. I try by the other way. Up. Is it working now?
No.
Still not. What can I change? You don't see anything?
We see a small stamp only on the top.
Okay. Okay, I'll try again. Sorry.
Yes.
So,
it's perfect. Perfect.
You can see perfectly.
Yeah.
Okay, cool. Okay. So sorry for that. Huh? So thank you, Mabel, for this introduction. And yes, today I'm going to try to present you the zebrafish as an innovative and emerging model for in vivo investigation of human and animal health diseases. So a few words to introduce myself. I'm a scientist working in infectiology of fish and rodent facility in the suburb of Paris. I don't know whether you can see the mouth, the pointer. I'm working here in INRAE campus close to Versailles in the University of Paris-Saclay. And in the unit where I am, we aim to produce and breed animals that are dedicated to infectiology experiments. And basically, we have the possibility to carry out experimental infection on animal in confined facility because we have BSL-2 and BSL-3 environment equipped with imaging instruments that allow us animal phenotyping. So our expertise are basically the development of in vivo model, conventional or axenic, and the setup of infectiology and inflammatory in vivo models that we characterize by a real-time three-dimensional imaging. So as Mabel mentioned, we are also part of the emerging structure, which is a national research infrastructure, who aim to develop models for in vivo investigation of infectious diseases that are of interest for human and animal health. So as I mentioned, you are really interested in development of infectiology and inflammatory diseases. And in the unit, we are working on rainbow trout and common carp. So we image the different fish with different setup. So we have access to photonic microscopy in BSL2 environment, or to an IVIS to register bioluminescent signal. So to illustrate the kind of images that we are producing, here on the left, you have pictures of live trout that has been imaged after infection with a virus encoding luciferase cascade, so that you can follow the spreading of the virus by monitoring the bioluminescent signal appearing at the level of the body, of the whole body of the trout. On the right, you have a 3D reconstruction of light sheet images we acquired from an entire carp of almost 4 centimeters long. And this is a new setup, a new technology that we developed to give access to 3D histological analysis of aquaculture fish diseases at cellular resolution. However, if you want to go deeper in the characterization of host pathogen interaction, then it's not that easy with such big sample. And for that reason, we work on a zebrafish as a model organism. So a few words about the zebrafish, because I don't know if all of you knows about this species. So this is a tropical freshwater fish originally found in India, in the Ganges River. And this is a small vertebrate that takes its name for the horizontal blue lines that are, you can see here, along the body. It presents a sexual dimorphism, as you can see that the female has a round belly, while the male has more golden appearance. And in its natural habitat, you can see that it's living in calm or stagnant water in shallow stream or ponds, and is able to deal with different, quite a large range of temperature and pH. He's omnivore, and he reproduces at the onset of the monsoon season, likely corresponding to the more abundant availability of food. It belongs to the Cyprinidae family and to the genus Danio, which counts almost 45 species. So there are really different advantages to use this model. And I have to say that maybe I like it too much. So probably you also have to consider that there are some drawbacks I won't mention. But one of it is that it presents a really rapid development. And by 24 hours, the embryo has already a defined body axis and rudimentary organs. So hatching occurs after 2 or 3 days and give rise to a free-swimming larvae. And the zebrafish reach sexual maturity around 3 months and can live on an average of 3 years and a half. The comparative genomic analysis highlight the fact that you have 70% of human genes that have at least one zebrafish ortholog. And this is kind of really amazing, because it means that this small vertebrate can be an interesting model to study ancestral biological process in a really small organism. We have access to genome editing tools and a ease of manipulation because of the small size. You can see here that the zebrafish larvae are fitting in 96 well plates which is really convenient to conduct high content, high throughput drug screening, for example. But over all these advantages, I have to say that one of the most important, to my point of view, is this optical transparency. So I choose to illustrate this by this movie, where you can see in real life the development of an egg and the embryogenesis. So the egg has been kept in the aquarium, and in fact, after fertilization in the water of the aquarium by the male, they remove the chorion, which is an envelope around the egg, and plug it with an agar plug here on the mounting on a cover slip, and image the development over time with a stereo microscope. So here you have the time going, and you can see that you see the development of the zebrafish embryo around the yolk sac. So here you see the tail, and here you see the head forming. And here at a certain point, around 24 hours, you will see the blood circulation, the blood flow, you can see here. So little by little, you see the development and the organogenesis. And here you can see the heart beating. Also, I guess that you notice that you have dark spots appearing, which correspond to the melanocytes that are covering the body little by little. So at a certain point, the fish will manage to detach and will give rise to this free swimming larvae that we were mentioning before. So I'll let you enjoy the movie. Up to the end, you will see that you will gently manage to detach. Okay. So based on the fact that we have so many genome editing tools, we also have access to a library of transgenic lines that are, serve as a reference for the study of, for example, immune system, because here you have this line presenting red neutrophils all over the body or green macrophages. And we also have access to tools to study vascular system and also nervous system and so on. So it compensates the lack of antibodies, and for that reason, as we miss all the tools that are available, for example, for mouse species, we used to extensively use this transgenic line to go deep in the description of biological process. All of these lines are kept and maintained in research centers that are able to deliver it to the scientific community. And here is an example of some movies that we took in real time to record, in fact, inflammatory processes based on the use of this transgenic larvae. So the transgenic is encoding red fluorescence. Sorry, it's not working here. Okay. So we are recording red fluorescence corresponding to the neutrophil in the brain of the zebrafish larvae after photoablation. So you see that we have access to the what we call the inflammatory response, because we can register the activation and recruitment of immune cells at the level of the brain, and also the regeneration of the tissue following the activity of this neutrophil. So as Mabel mentioned, in terms of ethical statement, the early stages of zebrafish are not protected as animals until the stage of being capable of independent feeding at around 7 days post-fertilization. Of course, it also relies on additional criteria, which are more morphologic, like the stage of development of the digestive tract and swim bladder, the ability to incorporate some food or the yolk consumption. But because of this reason, the zebrafish larvae is still considered as an alternative model to animal experimentation. So to summarize, we consider that it's really an innovative model to study in vivo processes, because it's cost effective. It allows you to have a short analysis time and to provide with high content, high throughput screening approaches. You have the ability to get this genome editing approach, as we say, to get live imaging, as we just saw in the previous slide, and also to model some diseases with which can be recapitulated with the immune system complexity. So for all of these reasons, zebrafish has been extensively used in developmental biology and more to model human and animal diseases. And now we have some tools that allow us to go deeper in the characterization of physiological aspects, also to study the behavior of the fish, to go with the screening of therapeutics, and to take advantage to the regenerative capacity of the fish to study after the inflammation, the cicatrization and regeneration of the tissues. So for the rest of my talk, I will just focus on viral infection, as my colleagues will further discuss about bacterial models. So as I told you, as I'm working in INRAE, we are much more interested in pathogen infection and therapeutic development in a One Health concept, meaning that we work in aquaculture diseases of farm and ornamental fish, but also on human health by the point of fish consumption and the study of zoonotic infectious diseases. We work in consideration of the environmental impact and try to develop therapeutics that are eco-friendly. So which pathogen for which questions is just to tell you that in fact zebrafish has been used historically to study fish pathogens, so RNA and DNA fish pathogen, as is indicated in this table. But later on, it's been also used successfully to study human viruses like herpes virus, VSV virus, chikungunya synbis, but also influenza. Also, zebrafish do not have any lung. So it's really a matter of having an in vivo model in between cell culture and mammalian system. And so it can be considered as a complex in vitro model, similarly as we consider now the organoids. So how do we process for the infectiology models? We used to microinject the pathogen, and you can see that similarly to what you are used to in mouse model, you can microinject the viral suspension in different regions. However, due to the small size of the guy, we cannot inject much more than nanoliters. So you have to go with high titer. Also, you will not need a lot of viral inoculum to be able to infect your larvae. We can also use the immersion, but it's not working in all the model for sure. So can we predict whether you will succeed to establish an infection model? In a way we can, because we know that zebrafish is able to deal with temperature coming from 25 to 33 degrees, meaning that your virus has to be able to replicate in this temperature range. And we also check whether the zebrafish is expressing viral receptors, for example. If it's not the case, then we go with genome editing tools and we make humanized zebrafish as we would do for the mouse model. An alternative will also be to use pseudotyped viruses if the idea is to study infectiology process for which you don't need the envelope of the virus. So this slide is just to illustrate the fact that based on the size of the zebrafish larvae, we can have confocal imaging of the whole body, but with a single cell resolution, meaning that during infectiology process, if you use, for example, recombinant viruses that are encoding reporter, you can go deep in the detail and see, as it is illustrated here, infected cells here that are red in the case of the synbis. For the chikungunya, they are red also. Here is an example of neurons, and you can see the axonal tract infected with herpes virus encoding GFP. And this is to illustrate the spreading of VSV-GFP. So in my lab, when we try to make a drug screening assay, the first step would be, as you may do in vitro system, to evaluate the toxicity of your compounds in the first step. So basically, we do it on some eggs that we collected in the aquarium. And we have we can obtain 100 eggs per female and per week. So we have access to a lot of samples, and we just collect them and put them in a multi-well and expose them to different drugs and at different concentration just by immersion. And over time, we register either a developmental defect or appearance of malformation, such as edema, abnormal body axis, and this kind of parameters. Once we made this acute toxicity assay, we go with a drug efficacy. And basically, we collect zebrafish larvae that can be transgenic or not. We infect them with a recombinant virus encoding fluorescent protein, and we challenge them with different drugs. After, we go with individual tracking of infection, like here, where it's illustrated, because of the fluorescence encoded by the virus, you can follow the spreading in the whole animal. And then we base our analysis on the appearance of clinical signs, on the survival rate of the larvae, on the big cell count when we go with image analysis. We can also make viral titration of from the larvae and go with transcriptomic and metabolomic approaches. So we end up with an integration of multi-parametric data set, which allow us to characterize the efficacy of the therapy. So when we speak about host-pathogen interaction, of course, we wonder what is the immune system of the zebrafish larvae. And this scheme just to mention that after 5 days post-fertilization, you have macrophages, neutrophils, and also T cells precursors, which allow you to study innate immune system. Basically, we used to make the microinjection of the pathogen at 3 days post-fertilization, when the larvae hatch and that you have access to a free-swimming larvae. So in terms of antiviral innate immunity, we contributed to show that whenever the virus infects the cells, the viral genome is recognized by the sensor of the immunity, like TLR, RLR, or TDR in the cells, in the fish, on the same way that what has been described in mammals. And this recognition evolved in the activation of TBK1 pathway and IRF and TNF-kappa-B transcription factor activation, which culminates with the production of type 1 interferon and pro-inflammatory cytokines. So after this production, we have an activation of the JAK-STAT pathway, and which evolved in the synthesis of interferon-stimulated genes, which are the main actors of the antiviral response. So in this really beautiful study of Jean-Pierre Levaux and Pierre Boudinot, there has been a comparison of the repertoire of ISGs activated in human and zebrafish in response to type 1 interferon treatment or viral infection. And then they identified a core ancestral ISG repertoire that is induced in the both species, commonly induced in the both species, and which constitute a nice candidate to identify biomarkers of viral diseases. So for the rest of my talk, I will illustrate what has been the impact of the zebrafish to model 2 different infectious processes and viral infection. I will first describe this study where we try to model the spring viremia of carp virus. So it's a rhabdovirus, and as you can see here, when you have infection of the carps, you have hemorrhages covering the body, and also you have infection and damage of hematopoietic organs. So when we inject this virus in the bloodstream of the zebrafish larvae, after a few days, you have hemorrhages that you can see here, and the larvae died from the infection. So by injecting some transgenic lines that express EGFP in the kidney tubules, we have been able to make some image analysis and to show that upon infection, you have also damage of the head kidney, which is the hematopoietic organ of the zebrafish. We managed to quantify this observation with the image analysis and shows that upon infection, you have a higher fragmentation rate of the kidney tubule as compared to control conditions. When we infect zebrafish larvae encoding a fluorescent GFP in the macrophage cell subpopulation, we managed to show that upon infection with SBCV, we destroyed like 80% of the macrophage population here that are imaged at the level of the tail of the larvae. We quantified also with image analysis, and we managed to show the correlation by making some RT-qPCR on the MPEG gene from the infected and controlled larvae. We also managed to show that this infection was leading to inflammatory response like IL-1 beta, expression of IL-1 beta, and expression of interferon type 1. So, of course, we are also interested in going further in developmental stages and go deeper in the details of the anatomy. And the problem is that over the time, of course, the tissue is thicker and you lose the transparency aspect. So how can we go deeper? So we have some tricks. For example, we use zebrafish mutants that are that do not have this melanocyte pigment. So they miss melanocyte, but also iridophore and xanthophore, depending on the mutant. And one of the well-known mutants in the zebrafish community is the Casper zebrafish, which is presented here. And you can see that it's basically almost transparent, which gives you access to a lot of information by 3D imaging of even juvenile zebrafish. The other possibility is to fix the tissue and to get it totally transparent. And my lab has been really involved in setting up this method for zebrafish species. And basically, you can see that by removing the pigment and making a homogenization of refraction index, you end up with here a totally transparent body. And basically, it gives you access to a lot of information. So this is just an illustration of what we have done. So we were interested once again to see the impact of SBCV on head kidney, on kind of juvenile zebrafish. And here is a kidney stroma in control and SBCV condition, infected condition. And you can see that the appearance is not the same. And here that you have the damage of the tissue. We correlate this result with classical histology data. And here is just to show you that by doing 3D reconstruction, we've been able to show in details the morphology of the kidney of infected and naive animals. So we take 3D images of a large stack of a pig sample, and then by 3D imaging, we have powerful histopathological analysis of the whole tissue. So I will finish my presentation with this illustration of the impact of zebrafish to study the neural invasion of 2 alpha viruses. And so this work was supposed to compare the infection and portal of entry of chikungunya and SINBIS, 2 viruses for which the lab of Jean-Pierre Levaux, with who I was collaborated at the time, was having recombinant viruses encoding EGFP or enteric acid, allowing us to track viral infected cells. So we were interested in defining the mode of entry and knowing whether it can occur through the vascular endothelium, through the trion earth using the infiltration by monocytes, or in the central nervous system, or access via peripheral nerves. So as I mentioned before, the use of zebrafish larvae has given access to a high resolution imaging, which has been really useful and suitable to describe the spreading mechanism of the virus. And based on the fact that we were having also the possibility to make some genome editing and some invalidation of cell population, we managed to show that chikungunya was using infection of endothelial cells to spread to the brain, to the central nervous system, while the SINBIS was preferentially spread via the axonal transport. So in this study, mostly focused on the chikungunya, the interest was also to be able to describe, in fact, what kind of infected, which kind of cells were targeted by the virus. So here it's a fixed sample, and you can see that by using different transgenic lines expressing fluorescence in hepatocyte, muscle cells, neurons, or ever, you can define basically in vivo which cells are infected, but without having to sample any tissue, but by live imaging of the whole larvae under confocal. They also have the ability to describe type 1 interferon response and to describe which were the interferon producing cells here at the level of the whole body. Also, the ability to cross interferon transgenic with neutrophil transgenic lines give access to this information and allow to precise that neutrophil were the interferon producing cells in this model. So I hope I managed to convince you that zebrafish can be a really interesting model to study in vivo animal and human health diseases, and that it can be at the interface in between in vitro and target species. So now I'd like to acknowledge the people with who I'm working in the team. Sarah Massad, in particular, Sarah Massad, who is working as a master student in our team, and Dimitri, who is responsible of the fish facility. The team of Pierre in the VIM unit, with who I was collaborating on a description of interferon responses in zebrafish larvae after viral infection. The team of Stéphane Biacchesi, with who I'm developing now a drug screening assay, as he is an expert in development of a reverse genetic tool for fish viruses. The Institut de la Vision for imaging, light sheet imaging of large sample, and all the members of the Frontier network, which is an international network with who is interested in innate immunity and inflammatory diseases. So I've finished and I'm open to answer to your question.
So thank you, Christelle. We don't have much question. We don't have any question yet. So I propose, so feel free to ask question during the talk, and then we can ask the question just after each talk, or gather everything and ask question at the end of the session. It's up to you. No question, so I propose Christelle to move on and continue with David presentation.
Don't have to stop share, maybe. Okay. So you still see my screen, right? Okay.
I'm sorry, my screen, I think.
Can you share it? Yes.
Yes, can you see my screen?
Yes, it is not in presentation mode.
So I'll take this time to introduce you, David.
Yes, that's perfect.
Yes. Okay. So David is a microbiologist who made his career in between Spain and France, and who explore fundamental and applied aspects of bacterial-host interaction. First, he's been involved in projects deciphering the virulent factor of different pathogenic bacteria, and now he focuses on understanding the microbiota-based function involved in the resistance to infection. For this aim, he used the Gnotobiotic zebrafish, which emerged as a promising animal model to analyze the role of different actors involved in this phenomenon. So today he will present the Notobiotic zebrafish model for the study of host-microbiota interactions.
Okay, so good morning, everybody, and thank you very much for this nice introduction. And also to the EAAP for inviting me to present or try to illustrate how the Notobiotic zebrafish model could be an interesting and useful model for the study of host microbiota interaction. So in fact, it is known that the microbiota have many beneficial effects for the host, independently of the host, I mean animals, human, insects, or even plants. Among these beneficial effects have been described that the microbiota could stimulate the growth of the host, as well as improving also nutritional level by helping the host to absorb specific nutrients on the development of physiological level, and also the development and stimulation of the immune system of the host. And in several cases also have been described that the microbiota could help the host to fight phase 2 disease. So there are still many gaps to try to explain mainly at mechanistical and ecological level how the microbiota can help the host. And for that, we need to perform these studies, the study of the role of the microbiota under an in vivo context. But there are many in vivo models available, and with the advantages and the disadvantages. In fact, in one way, we can find the mammal systems that they are highly pertinent in terms of complexity at development, physiology, and immunology level, and even at microbiota composition level. But sometimes this kind of models does not allow to perform the high throughput studies due to, in several cases, they are quite hard to manipulate. And the other way, we have the invertebrate systems that they are like C. elegans or the Drosophila that allows to perform a very high throughput studies due to mainly their small size and the short generation time, but they are quite limited in terms of complexity, and sometimes they are quite far from what we are interested in, I mean, in the context of the human or animal research. And among all these different animal models, zebrafish emerged as a very interesting and promising model because of the different advantage of this model that Christelle already present very well in her nice talk. But just I will try to explain them so quick. The short generation time, because the life cycle of this animal are quite quick comparing with the other animal models like mammals. There are also many genomic tools available to create mutants or transgenic lines. Also, as Christelle illustrates very well in her presentation, we can perform very nice non-invasive imaging on the zebrafish, thanks to the transparency of the zebrafish mainly at larva stage. We can also perform a behavioral testing, and we can obtain quite easily germ-free individuals. And it's in this aspect in which I'm going to focus my presentation today, because to obtain germ-free fish is the first step to obtain later the notobiotic individuals. Notobiotic, it means from Greek, notos means noun, and biotic life, so noun life. And the idea is usually in the conventional cases, the embryos of the zebrafish are surrounded, let's say contaminated by the own microbiota in the surface of the eggs. And once that they hatch, part of this microorganism will colonize and develop their own natural microbiota of the conventional fish. But thanks to the ex utero or in ovo development of the zebrafish, we can sterilize the eggs to remove all the bacteria associated to the surface. And by keeping these individuals under sterile condition, we can obtain germ-free zebrafish. And it's with these germ-free zebrafish that we can obtain the notobiotic individuals by exposing them to a specific bacteria. It could be just a single bacterium, just to see a kind of probiotic or even a pathogen, or to a consortium of bacteria to try to mimic or the natural microbiota of the host. And in fact, this model, the germ-free and the notobiotic models, are being used more and more in research in the last 20 years to explore different aspects. In one way, for example, it has been analyzed by the team of Kate Gilliman at the University of Oregon, but it's not the only team, but they did a very nice research about how the microbiota is involved in the host development. And that they saw is that in one way, the microbiota is essential for in the epithelial differentiation of the host. Here we have a picture of one of the work in which they illustrate how the conventional zebrafish have a correct or high level of alkaline phosphatase activity in the gut, which is significantly reduced in the germ-free fish. And interestingly, when we reconventionalize the germ-free fish with specific microorganisms, we can restore this alkaline phosphatase activity. Also, they have demonstrated that microbiota is involved in gut motility. So the presence of the microorganism in the conventional or reconventionalized fish stimulate the peristaltic movement of the intestine. And they have shown also how the microbiota can stimulate the epithelial proliferation, in which they compare how, in the case of conventionally raised fish, they have a higher amount of proliferating cells comparing to the germ-free raised fish. So this is the case in the host development role of the microbiota, but it has been also largely analyzed how the microbiota can affect the development of the innate immune response of zebrafish. Just an example, a global example, it has been described that the presence of the microbiota could increase the amount of different parameters associated with the immunological response, like the neutrophil numbers and speed, also the pro-inflammatory mRNAs, or the alkaline phosphatase activity, as I have shown you previously. In the context of the infectious disease, zebrafish has been also largely used, Here you have just a snapshot of the different pathogens that have been tested and validated in the Zebrafish model. Here are mainly human pathogens like Pseudomonas aeruginosa interacting with neutrophils zebrafish, so the infection, successful infection of Listeria monocytogenes or Staph aureus, among others. But I'm not going to describe too much about this point because I know that Vincenzo Torraca prepared a very nice talk about this topic, so I strongly recommend you to attend to his talk. But in the context of the infectious disease, the Notobiotic Zebrafish model emerged as a very promising tool. Why? Because this model, the germ-free Notobiotic model, could help us to understand what is the role of the different actors involved in this process. I mean, the host, the zebrafish, the pathogenic bacterium, but also the non-pathogenic bacterium that could be present in the natural microbiota or just exogenously added bacteria that could be probiotic candidates. And it's based in this model that our work in the unit started a long time ago with this nice work driven by Olaya Rendueles, in which she described a new Zebrafish model of our intestinal pathogen colonization, in which he was interested in the fish pathogen Edwardsella ictaluri. It's a fish pathogen that can infect different fish species. And she demonstrated that this pathogen, the Edwardsella ictaluri, was able to efficiently infect and kills the germ-free as well as the conventional raised zebrafish larvae after exposition of this larvae by immersion by bath, adding the bacterium, the pathogen, to the water. They even demonstrate that this bacterium was efficiently colonizing different tissues of the zebrafish, of the infected zebrafish, as you can see in this picture, where we have the red spots in the gut. The red spots corresponds to the Edwardsella ictaluri, and the high concentration of this bacterium in the gut and also in the head close to the gills. But what is interesting was that they have observed that Edwardsella ictaluri can infect the germ-free zebrafish. They analyze how the different probiotic bacteria or candidate bacterium could protect or not against infection. And that we can see in this graph is that when we monoreconventionalize the germ-free fish with a non-pathogenic E. coli strain before the infection, we see that this bacterium alone is not able to protect against the infection, and the fish die at a similar level than the germ-free fish. But interestingly, the same strain carrying the F plasmid, responsible of the development of a strong biofilm or highly adhesion of this bacterium, we can observe an increase on the survival of the fish exposed to the Edwardsella ictaluri. So that suggests we observe also the same phenomenon in the case of other highly adherent E. coli strain, and which suggests that the probiotic adhesion properties plays a key role in the protection against the Edwardsella ictaluri infection. And this work validates the Zebrafish model as a very interesting model for the study of potential probiotics candidates. But we wanted to go first about this topic, and in fact here we validated that in the previous work we validated that the probiotic candidates could protect the zebrafish against infection, but we wanted to explore what happens in the case of the natural microbiota of the host, because in the microbiota we have mixed of bacterial communities that they are continuously interacting between them in a dynamic way, and they are also interacting with the potential opportunistic pathogens that could be present in the microbiota, but also with external pathogens that could try to colonize and develop the infection. And it is known, it has been described that in several cases, the microbiota, the presence of microbiota, avoid the colonization and the development of the infection. But it is not well known how it's happening, and there are many hypotheses that have been proposed, like a barrier effect, just the fact that there are a mass of bacteria colonizing the tissue will avoid the efficient colonization of the host. Also, a membership effect is not the fact that there are many bacteria present, but there are several bacteria that will be the key for the protection against infection. The other possibility is the social structure, which means that the interactions between some of these bacteria present in the microbiota will protect against the infection. Or finally, it has been proposed also that since it is not the microbiota stimulate the immune response of the host, some of these bacteria present in the microbiota will help to develop a better or more efficient response against infection. It could be one of these hypotheses or all of them. So to explore this phenomenon, we started working with another fish pathogen, Bacterium columnare, which is a gram-negative bacterium that infects many different fish species, from cold to warm water fish species, I mean from trout to tilapia and even the zebrafish. And that we observe in the lab is that whereas the germ-free fish are highly sensitive to the infection of Bacterium columnare, the conventional fish, those that they carry their own microbiota, are highly resistant to infection. The Bacterium columnare is not able to kill this conventional fish, which suggests a very exciting result, because it suggests that the microbiota associated to the host, the conventional host, could provide this protection against infection. So to try to explore more in detail this phenomenon, and just to try to make a long story short, we isolate different microorganisms coming from the conventional zebrafish, a total of 10 different bacteria, to reconventionalize the germ-free fish with this bacteria before the infection. And that we observed first was that when we reconventionalize with a mixture of these 10 bacteria, that we call it mix 10, before the infection, we observed that this bacterial consortium was able to protect the zebrafish at similar level that we observed in the case of the conventional fish. To try to see if any of the bacterium forming this mixed 10 consortium is more important than the others for the protection phenotype, we mono-reconventionalize also the germ-free fish with each species before the infection, and we observed that from the 10 bacteria, only one was able to protect alone, this one that was identified as Christobacterium. But another very exciting, interesting result was that whereas the other 9 bacteria alone were not able to protect the zebrafish against infection, when we reconventionalize the germ-free fish with a mix of these non-protective bacteria, we restore the protectional levels of the conventional zebrafish. So these results suggest that probably we are in front of 2 different protection scenarios. So in one way, we have the protection driven by Christobacterium. It's an individual protection, highly robust, because we don't need a very high amount of Christobacterium to drive this protection. And it's also robust because it still protects a high concentration of Bacterium columnare. And in other way, we have the group protection driven by mix 9, which is less robust in terms of variability, but still interesting because we can explore which are the different interaction between the bacteria to provide this protection in vivo. So now we are working to try to identify which are the molecular and the ecological mechanisms involved in this protection. That we explore was if the immune response was involved or not in this protection. Since it is known that the fact that the bacterium, the fish that they are not exposed to bacterium have a less efficient immune response. So for that, from the individuals that we well known that they are protected or not to infection, we explore 2 different interleukins. Here we are showing you just 2 examples of the immune response answers that we have analyzed. And in fact, the first thing that we have observed in this case, that's in the case of the germ-free fish, we saw a strong induction of gut inflammatory response after infection, as you can see for the EL1 beta and EL22 interleukins. And interestingly, we observed also in the case of those fish that they are protected and to the infection, we have a downregulation of this immune response after infection, which is so promising results. But then when we include a control bacterial consortium that was not able to protect, like for example, this mixed Furber bacterial consortium, we observe that this downregulation of the inflammatory response is not specific. That means that it's happening in the protective and non-protective bacterial consortiums. So it seems that the protection that we observe in our model is not based on immunomodulation. And so we continue exploring to try to identify which are the factors involved in this protective phenotype. One of the questions just before finish, one of the questions that we ask ourselves is, okay, so we identify a bacterium that is able to protect the Zebrafish larvae against Bacterium columnar infection. So could this bacterium, Christobacterium, act like a broad spectrum probiotic candidate? So for that, we explore the ability to protect of this bacterium in different contexts. In one way, we test if this bacterium is able to protect just adult conventional Zebrafish, though that they carry their own microbiota. This experiment were done in collaboration with Remit Rigaudot in the IEAAP facilities at INRAE. And that we observe is that whereas the conventional adult Zebrafish were sensitive to the infection of Bacterium columnare, when we expose the conventional Zebrafish to Christobacterium before infection, just 48 hours before the infection, we see how we have an increase in the survival of Zebrafish comparing to those that they were not exposed to Christobacterium, which suggests that Christobacterium is able to protect adult Zebrafish. But to try to go deeper in the ability to protect of this bacterium, we explore if this microorganism was able also to protect other fish species. In this case, we were focused in the rainbow trout, which is economically interesting fish species that is produced in fish farms worldwide. And for that, we developed a new rainbow trout, the germ-free rainbow trout model to explore the ability of different probiotic candidates to protect against infection. And as you can see in this graph, whereas the germ-free rainbow trout fish are highly sensitive to the infection, those that they were exposed also to Christobacterium were also highly resistant to the infection of Bacterium columnare in the similar way that we observe in the case of the Zebrafish. So to answer to our question, yes, it seems that the Christobacterium that we have isolated from the conventional Zebrafish larvae could act as a potential interesting probiotic candidate in different in vivo context. So I see that the time goes really fast. So just to conclude, I hope that with my talk, I convince you that the Notobiotic Zebrafish model is a very interesting and promising model for the study of the role of microbiota in an in vivo context, taking in consideration all the actors that are involved in this phenomenon, from the host, the community, the microbiota, and also the pathogens. And in fact, we have demonstrated that by mining the fish microbiota, we can identify the key contributors to disease resistance. And this model, the germ-free and the notobiotic model, is a well-adapted experimental model for the study of infections, but also of probiosis. So before to finish, I would like to thank all the people involved in these projects. Mainly the people from the unit, genetics of biofilm unit, to Jean-Marc Guigaud, the head of the unit, who gave me the opportunity to participate and develop different parts of these nice projects, and also the formers and the current members that they are working on this project. Also the different collaborators in the Institut Pasteur from the macrophage and development of immunity author group platform that they provide us with their deep knowledge about the Zebrafish, and also the external collaborators, with special thanks to the people from INRAE that they help us continuously with their knowledge about the pathogenesis of the Zebrafish and the fish pathogenesis in general, and also to all of you for your attention. And now I will be happy to answer your questions. Thank you.
So thank you very much, David, for this very nice presentation with very didactic and nice pictures. There is no question.
So I have one or two if no
okay,
one.
So I let you the.
Okay. So the first one is that at the first part of your presentation, you mentioned that microbiota is required for epithelial differentiation. Can you just elaborate a bit and tell us about the mechanisms that are involved?
So about the mechanisms, I think I have to read again this paper because it's an old paper, but I think that they did not describe very well how is involved the microbiota in the development. So I have not in mind the details of this work, so I'm sorry, I cannot give you more details.
Okay, so the second question is that do you know about viral infection models that can be of interest to challenge, the effect of your probiotic in the protection of the intestinal disease?
So I think that it's the Notobiotic model is a promising tool also for the study of viral infections, because there are some virus that could infect, could develop the infection by the intestinal, let's say, colonization. If it is not the good word, but it's a promising tool for sure, and also to see if what is the role of the presence of the microbiota, just comparing the conventional and the germ-free fish, and then after infection with the several virus. So I think that is a promising tool that we can use. We never explored it yet, but it's something that we can explore for sure.
Okay. You know that we are working on the establishment of inflammatory models based on chemical application. And in these models, in fact, the conventional zebrafish died even more than than the germ-free animals that are obtained at this moment with the antibiotic treatment. So meaning that the microbiota can be even damageable for the phenotype we are observing in a way. So we have to reproduce this data of the literature, but I will be happy to discuss more about that with you.
I will be happy.
The last question was that, okay, you define a probiotic and with really beautiful effect that can be transferred to trout model. But let's imagine that you would like to apply it in real farm aquaculture. How can you deliver it? And what will be your expectation in terms of protection in this species where I don't know whether we have some knowledge about the composition of the microbiota in the farming installation?
So it's a very nice question, and it's the next step that we are exploring with this bacterium. We want to see at what level this bacterium can protect real life, let's say, the farming fish in the real life conditions. That we know is that at least for the trout, this probiotic candidate could well colonize the gut of the host. For the delivery, there are many ideas. For the moment that we did until now is just by adding the bacterium into the water, but something that we cannot imagine in the big pools of fish. But another delivery could be just by including this microorganism in the feed, and just see if it works or not.
Okay, but in the feed, is there any microbiota that can interfere with the effect of your probiotic?
It's hard to say that there is microbiota associated to the feed, because sometimes we are using dry food. So in this case, the microbiota associated to this food is usually the environmental microbiota of the specific installation. So it's for that it's quite hard to say that it's a microbiota interacting with the potential probiotics.
Okay. Thank you, David.
Thank you.
I have just a question coming back to the role of microbiota and the facilitating role or inhibiting role of microbiota in regards to viral infections. I was thinking about some very nice work, a set of works that was done on enteroviruses, but it was in mammals, showing that some specific bacteria from the commensal microbiota was able to facilitate enterovirus infections. And this was done mostly in rodent and mice. And do you think that this can be something that could be addressed using your model with colonization and specific colonization and things like that? And how could it be complementary to what is done in rodent? So I think or the advantage and the limitation finally of your system in comparison with what is done in rodent?
Yeah, so to answer to the first question, yes, it's something that we can address with this model, with the Notobiotic models, because we can follow how the different taxonomic groups that we have included in our Zebrafish, the behavior or the amount of these taxonomic groups that we have added to the Zebrafish before infection, how they change after infections. And for sure, we can identify those that they are manipulated or just stimulated or decreased due to the infection. So yes, we can address this kind of studies. And for the second question, can you remind me, please? It was
the question was regarding the limitation of your of the Zebrafish systems with the three partners, microbiota, virus or pathogen, and then and the Zebrafish versus the rodent as host. So I think that there might be some limitation for both. So of course we saw that technically it's much more convenient to work on Zebrafish and much easier. But regarding other mechanism for physiopathology and things like that, and it could include also adhesive molecule, or are they very close or not from what is present in mammals, for instance?
There are two main limitations that I have now in mind. So one of the limitations is the temperature in which we are raising our individuals. We can go until 32, 33 degrees. But comparing with mammals, that usually for the pathogens, we need to raise the 37 to, in several cases, even 40 degrees. So we are far from this ideal optimal temperature for the pathogens. And then also for the microbiota composition. So in fact, the microbiota composition of the zebrafish and mammals is quite different. It's clearly different. In fact, in the case of the mammals, they are dominated mainly by Bacteroidetes and also Firmicutes. And in the case of the zebrafish, it's mainly Proteobacteria. So it's so different. But and I know that there are several works that have been done just to obtain humanized zebrafish microbiota, but with not very good success. And in fact, apparently there is a strong effect of the host for the successful colonization of the bacteria. I think that there are a lot of work that we can do to try to improve these results, but one of the limitations also is that some of the microorganisms present in the microbiota of humans or mammals are anaerobes, extreme anaerobes. And in the case of the zebrafish, at least at larvae stage, it's very difficult to find anaerobic environments in the gut because it's quite microaerophilic environments.
Okay, thank you.
You're welcome.
So I think we can move on and go to the presentation of Vincenzo. Is it okay?
Yes.
Yes. So it's my pleasure to introduce Vincenzo. So he's an assistant professor at the London School of Hygiene and Tropical Medicine in London. He did his PhD at Leiden University, where he worked on a zebrafish model to study host-pathogen interaction, focusing on Mycobacteria and Salmonella infection. He was then awarded the Marie Curie Fellowship to move to Imperial College, where he developed a zebrafish Shigella model. He is now exploring this model further in London to understand how Shigella evolved antimicrobial resistance during the infection of the host. The title of this presentation will be Zebrafish Infection from Pathogenesis to Cell Biology. Thank you, Vincenzo.
Thank you, Christelle, for the introduction. My presentation is going to be on how revolutionary the zebrafish model has really been for us to understand the cell biology of infection. So the interactions at the single cell level between immune cells and, in my case, bacterial pathogens. Actually, Christelle beautifully introduced this, so I can fly on the introductory slides. I actually have picked the same video that Christelle presented earlier, just to exemplify how useful and how strong, powerful the optical transparency of the zebrafish model is. And I think this is what attracted initially developmental biologists and then immunologists to actually watch how host pathogen interactions happen live. Also, another thing I wanted to add is that the development is incredibly fast. So the macrophage and the neutrophils, which are the cells I'm interested in, are developed by 24 to 36 hours post-fertilization. So already from that time, the zebrafish model is useful for this kind of research. And then, as we just said, it actually really condensed together the power of being an animal model, which is a vertebrate model, with different route of infections and blood circulation and all these vertebrate-like structures and organs, together with actually tools that normally we are more accustomed to using in vertebrate models or in vitro models. And definitely the most important aspects are the optical transparency that actually combines very well with genetic manipulation, so that we are able to make mutant and transgenic lines and sort of Frankenstein mutants all the time, like we like to do. Just here, the genetic toolkit is it's continuously expanding, and the zebrafish really is coping very well with the evolution of technology. So, for example, in the past, we used to use a lot transient technologies, which are still very useful, like overexpression of RNA and morpholine knockdowns. But now, as the technologies are expanding, people use more and more these stable tools. For example, CRISPR-Cas, the zebrafish really literally looks like being designed for exploring optical tools to make stable lines. And just an example of how these look like, this is a transparent zebrafish larva with all these macrophages moving around. And so you can appreciate how much information a video like this can actually give. And these are macrophages that were not challenged, and it shows that macrophages recirculate and random patrol quite a lot. And then these instead are macrophages in red that actually are directionally moving towards a locally injected mycobacteria in green. By watching this movie over and over and for a longer time frame of the movie, you can also start to generate a lot of different hypotheses. For example, you can see that by 48 hours, the bacteria have not been cleared at all. And actually, you can also see macrophage and neutrophils in this area moving the green fluorescent of the bacteria away from the field. And so, for example, this generates a lot of information about reverse migration and the difficulty of clearing mycobacterial infections. As also David has introduced, over the time, scientists have collected a battery of different tools and assays that can be done in the zebrafish model to explore different aspects of the host pathogen interaction at the single cell level. And this is sort of a menu of different tests and analysis that can be performed very well. And for example, infections with Salmonella have been very informative to understand how macrophages are being reprogrammed in terms of metabolism, and they express different things when they are infected. You can also do analysis of the immune cells at the whole population level. For example, here was shown that during Salmonella infection, the number of neutrophils drops very, very rapidly. And after that, there is a trigger called emergency granulopoiesis that brings the number of neutrophils up again in large number to cope with the infection. You can zoom even further. For example, here you can look at single proteins from the host. These are septins, and how they interact with an individual bacterium and form a cage around them to actually stop the bacteria to grow. And I think this was also presented before by Christelle. Another example is how Listeria moves from one cell to another by exploiting the actin cytoskeleton of the host to be propelled from one cell to another. Another examples here is how actually the zebrafish also is contributing new ways that we can actually approach host pathogen defense against pathogens. So, for example, here the zebrafish was infected with the antibiotic resistant Shigella, and it was and we used a predatory bacterium, Dello vibrio, that feeds on Shigella to actually clear the infection with an antimicrobial resistant threat. Other examples of macrophage reprogramming, for example, Burkholderia only works parasitizes a subset of macrophages that become IL-1 beta positive. Macrophages here, after infection with the Mycobacterium leprae, they basically produce toxic compound that can lead to the neuropathy and the damage that leprae does actually to the neuron. So it's not really a direct effect of the of leprae on the neurons, but actually is a side effect of the change the metabolism of the macrophages. Another friend of mine works instead on the immunological bottleneck that actually develops in Staphylococcus infections. And here, for example, by injecting with Staphylococcus of two different colors, he showed that actually the two colors never mix, and there are phagosomes inside neutrophils that either carry one or the other fluorescence, and that's because there is such a strong bottleneck, and these focal puncti actually derive from one single bacterium that replicates, and this is at the base of, for example, the focal abscesses that actually Staphylococcus can form. And of course, my favorite is Mycobacterium marinum, and for example, people have substantially used mycobacterial infection to study the cell biology on how the cells actually cope with bacterial pathogens. And here it was shown that, for example, autophagy, which is a physiological mechanism to keep the cell clean and tidy, and a starvation mechanism can also be used to recollect bacteria that escape outside the phagosomes to actually clear the pathogen. Mycobacteria are actually infection in zebrafish are actually a success story. And so over the years, people have really used the zebrafish model to understand how the granuloma, which is the typical hallmark of mycobacterial disease, is actually formed, and how mycobacteria actually orchestrate the recruitment of macrophages and the dissemination of the infection via other macrophages. And another success story that I would like to share is actually the use of the zebrafish model to study strictly human bacterial pathogens, such as Shigella, which is a human-adapted pathoviral E. coli. And actually here we can see that the zebrafish has contributed to us understanding that different cell types actually deal with infection in a very different way. So this is a movie, and here we have an infected epithelial cell fully packed with the bacteria, and you can see that they are not able to really cope with the bacteria very well, and the green fluorescent builds up over time. However, the neutrophils in red around, they're very excited about the infection, and as soon as the cell will die, and it will happen in a few seconds, you will see how quickly they're able to scavenge all the bacteria, and actually the neutrophils don't die. So it's happening now, and in few minutes you can see that the neutrophil is able to cope with the infection. So we can actually understand how different cells behave differently. So I would like you to remember this, and actually this is Shigella flexneri, and it will become important later. So just to recap in terms of Shigella, where I would like to focus on the rest of my presentation. These are human-adapted pathoviral E. coli, so they are virulent for humans because they have a virulence plasmid that makes them able to cause a diarrheal disease because they invade epithelial cells. This is still a very important threat, 165 million cases every year and 600,000 deaths. And in particular, is also in the top 12 list of bacteria that have that need to be addressed because of the continuous evolution of antimicrobial resistance. And in the zebrafish model, one of the systems I use most is actually the injection of Shigella into the hindbrain, because this model is immediately accessible for imaging, but also because we can watch how macrophage and neutrophils get recruited and they interact with infection, and also because we can score the infection with different easy systems such as the bacterial load and or, for example, with survival assays. And here, as an example, this is a treatment with an antibiotic to show that actually Shigella would be contained. And so you can imagine this can be done with different hosts and pathogens, mutants, to actually address different aspects of the disease. When I joined the Mostovi lab at Imperial College, they were working on Shigella flexneri. But however, having a look at the map of the distribution of Shigella disease, I found that actually Shigella sonnei has recently become the dominant cause of dysentery worldwide, and the countries in red now report more cases of flexneri, of sonnei than of flexneri. And so for this reason, I wanted to use the zebrafish model to see actually if we can do comparative analysis between different strains of Shigella to actually understand how these epidemiological trends develop. And so, for example, here I found that using the survival assays, the Shigella sonnei is dramatically more virulent than Shigella flexneri. Here, 80% of the fish die in 3 days, while for flexneri only 20%. And this goes together with an increased bacterial load and with more dissemination, more inflammation, the whole package. And so over the years, I figured out that actually what is unique of Shigella sonnei is its antigen. It is rather special. And if we remove the antigen, the bacteria completely become virulent. However, the antigen, what it does, it's actually it's really important to cope with neutrophils. And in fact, if we remove neutrophils, as we can do in the zebrafish model, then the fish can die also in presence of an antigen mutant. And so we turned to live imaging, and we found that in fact, while Shigella sonnei actually is able to resist into neutrophils and it recruit neutrophils, it actually really acts like a sink. The localized infection here will exhaust the neutrophil population quite quickly, while the antigen mutant doesn't do that. And the same way also Shigella flexneri, as I showed the video before, doesn't do that, and it's not surviving in neutrophils. What we found is special for Shigella sonnei is that they are actually able to adapt, thanks to their antigen, to the acidic environment that they actually have into neutrophil phagosomes. And here we can see that, for example, there is an advantage of the wild-type bacteria to grow also in an acidic pH compared to mutant bacteria. And so the bottom line is that Shigella sonnei antigen is driving the expansion in the human population of Shigella sonnei, because actually it allows them to also survive to neutrophils, like while Shigella flexneri is not able to do that. And so my recent work actually focuses on trying to understand other epidemiologically relevant questions on Shigella distribution. And for example, I'm trying to understand if the zebrafish model can be illuminative to predict how the antimicrobial resistance is evolving and spreading in Shigella sonnei. And these are countries that already report at least one antibiotic resistance to one of the 3 antibiotics that we still use to treat Shigella nowadays. And another important epidemiological problem with Shigella is the emergence of Shigella persistent infection. So everybody thinks that Shigella causes the real disease that is cleared within a few days. So it's an acute self-limiting infection. However, new data collected in the UK and in other countries have shown that a lot of people actually have persistent infections with low bacterial load that are present for weeks or even months. And this is an important problem because the infection can be reactivated later on in life and cause the disease again, but also because it can be transmitted very easily via asymptomatic carriage. And so I would like to understand if the zebrafish model also can illuminate on Shigella persistent infection, which is really poorly understood. And so I set up a model to study Shigella persistent infection in zebrafish. And here I found that actually the infection in most of the larvae, in fact, expands with an initial acute phase, but then the bacteria are cleared at a steady pace for several days. And after a certain point, actually, the bacteria are no further cleared by the host. And actually, in this case, the bacterial load that I use is very small. It's in the order of 1,000 bacteria only, and still not all the bacteria can be cleared. And the link I have between persistent infection and antimicrobial resistant, my other interest, is that actually you can establish persistent infection even during treatment with an antibiotic. This is an aldexic acid, and you can see that while the bacteria do not proliferate, they don't go into acute phase, also they are not cleared for multiple days. This happens also when the bacteria are established as a persistent population first, before even seeing an antibiotic. So for example, in this case, this group was not treated with an antibiotic, but the bacteria were allowed to become persistent. And then if you treat them with an antibiotic for 24 hours, the antibiotic is not able to clear the infection. While you can appreciate that if you do the same in the acute phase, the bacterial load significantly drops with treatment with the antibiotic. And this phenomenon is called antibiotic tolerance, and is considered to be an important step towards evolution of AMR. Another interesting finding is that while I spoke about neutrophils before, it looks like that persistent infection really hides inside macrophages. So the few bacteria that are left are together with a lot of bacterial debris, bacteria that are not culturable, inside this small discrete number of macrophages. This is a movie showing, I'll start this again. This is a movie showing that actually the association of some Shigella with the macrophage can be quite stable for multiple hours. And while we are normally accustomed to actually see either the macrophage dying or the bacteria being killed, in this case, for multiple hours, you can see the macrophage still being happy and moving around, and the bacterial load and the fluorescence of the bacteria are not really changing over time. Another interesting connection between persistent infection and antimicrobial resistance is also that I found not only that the bacteria become tolerant to the antibiotic, so they do not die when treated with the antibiotic, they also change the rate at which they can accumulate mutations. And this is because some genes that are involved in the mutation frequency, such as mutase, which is a DNA repair mechanism, are actually suppressed in vivo. And actually, I managed to quantify that, in fact, during persistent infection, the bacteria can evolve real mutations at a certain frequency. Now, this number doesn't seem very intimidating, it's small, but actually in vitro, this antimicrobial resistance occur at a frequency of 10 to the minus 9 or 10 to the minus 12. So this is at least 1,000-fold higher than what we normally are accustomed to see. So in my hypothesis, basically, a persistent infection can facilitate AMR in two ways. It can sustain antibiotic tolerance by making the bacteria to survive treatment with an antibiotic, and at the same time, by suppressing the DNA repair mechanism, it allows them to accumulate mutations that will make them real antibiotic resistant. So the overarching idea I have is that the zebrafish model not only can really inform on the cellular biology of infection from the single bacterium to the whole organism level, but eventually will also be able to illuminate on studying, approaching research in infectious diseases at a whole population level. And population level doesn't only mean the host population, but also the bacterial population. And that's because even if we think of Shigella, there is different species, and within each species there is a lot of different clinical isolates that separate in different lineages and sublineages, and these can be more or less successful in a host. And so, for example, just to conclude, the antigen that is important for several things, it's also dictating the establishment of persistent infection in Shigella sonnei. And actually, when we explore variants that naturally occur of Shigella or antigens, for example, Flexneria has different antigens, we can see that the level and the efficiency of persistence is different in the zebrafish model. And actually, there is a strong correlation between the level of persistence that we see in zebrafish and the epidemiological incidence of cases of persistence of these different antigen variants in the human population. And so this indicates that zebrafish research can really illuminate clinically relevant findings. And just to conclude, this is on the pathogen side, but on the host side also, the zebrafish will not stop to surprise. And for example, this year, some people have set up a system which is called mic drop. And after this, I will drop also my microphone. And this means multiplexed intermixed CRISPR droplets. And basically, they pack individual CRISPR-Cas mutagenesis reagents in single droplets, and then each cell of the zebrafish, its egg, receives one droplet, and so we will develop into a single mutant for a specific gene. And so we can imagine to inject each of these zebrafish larvae afterwards with bacteria to actually see how the different mutations that the fish carry will actually affect the establishment of the infection. And with this, I would like to thank you all. And if there are any questions, I'll be happy to take them. Thank you.
Thank you very much, Vincenzo, for this presentation. So you clearly show how nicely you can nicely acquire information on immune cells and the pathogen interaction. Also, it's incredible.
We'll start with a question, maybe. Yes. So you show that you have a persistence in zebrafish larvae. So maybe I missed some of the details in your presentation, but can you remember me at which stage you inject the larvae?
So we inject them at 3 days post fertilization. So this is 3 days post fertilization.
Okay.
And then 144 hours is actually 6 days post infection. So that's by the time they're 6, 7, 8, 9 days. Yeah.
And can you predict whether you will have the persistence if you inject in another place than the brain? Because
So,
I guess that the brain is not the natural route of infection. So
what do you mean? Can I predict?
Yes, I mean, I don't know by which way humans are infected, but I guess that you select the brain intracerebral injection because it's more convenient or for reproducibility. Yeah. But can you predict that the phenotype of the persistence in terms of tissue tropism or host response would be different if you inject in another site?
Okay. Yeah. Well, so I've seen persistent infection also to cure in the systemic infections, so they can still form. The only thing is that the infection goes a little bit wilder when you inject into this into circulation. So you actually really can inject a very, very little amount of bacteria. Otherwise, many will die. Many fish. While the fish generally copes relatively better with injection into the hindbrain because it's contained. And so we have many more larvae that establish persistent infection with a dose that actually is more how do you say easily controlled, so all the fish actually really get the same. When I noticed that, for example, if you try to inject less than 500 bacteria, then there is quite a large spread of the amount of bacteria that they get. So the hindbrain model is a very good compromise for a very reproducible system. They're actually
and to keep the larvae alive so that you can follow the phenotype, right?
Yeah. Another interesting thing I wanted to say is that so with this dose, which is about 1,000 bacteria here, we actually get easily 90% of the fish establishing persistent infection, and the other 10% essentially die. So they develop an acute infection from which they do not survive. If you have a higher amount of bacteria, so for example 8,000, then 90% of fish die, so they do not recover from the acute phase, and the remaining 10% establish persistence. So we can basically study one or the other depending on the initial bacterial load.
I guess it's just a question of microbiota, you know. No, I'm kidding. Just for
okay. No, actually, it's very interesting that you mentioned the microbiome, because obviously that's something I haven't explored yet, but it would be really interesting to see what the role of the microbiome is in the establishment of these, because I think in humans it might play a very important role by making So one hypothesis I have is that in humans, in humans, people have shown that macrophages, for example, do not cope very well with Shigella. They normally die, but the macrophages that reside in the gut actually are much better trained to cope with with infections, and they do survive relatively well several insults. So I think that the missing link here is that maybe those macrophages are very special and they could harbor persistent infection more likely.
Okay, and there you come with my second question, which was that you mentioned that you are studying host interaction with the bacteria while you inject in the in-brain. So how what the origin of the immune cells that are coming? Don't you think that the macrophages that are interacted at the one of the brain, like the microglia, or do you think that they come from the periphery?
Yeah, so we see that they mostly come from the periphery. So they are recruited, so they derive from monocytes. There is obviously microglia there, but I think that actually the persistent infection is established in some that were recruited from the monocytes, because we see that the initial cohorts of cells that are locally present actually die after infection, and then there is second events of recollection of bacterial debris and cell debris by newly recruited immune cells. And definitely in the bloodstream also it happens in monocytes-derived macrophages.
And do you have neuronal damage because of this toxicity?
It would be fantastic to actually have a look. I'm pretty sure there would be. These fish surprisingly swim very well. I haven't seen. I mean, they seem a little bit lethargic, but they swim. They're fine, and they carry this persistent infection. But it would be really fantastic to have a look.
An idea, yes. Okay, and did you try on juvenile zebrafish to see whether the maturity of the immune system can modulate the response or give something different?
I haven't tried, but I'm really looking forward to doing those experiments. Unfortunately, for ethical reasons, that's as far as we can go. It's 9 days at the moment. I would really love to do that.
Okay.
Yeah.
Thank you, Vincenzo.
Thank you. Thank you.
I have a question. Hi.
Hey.
Very nice talks. Thank you. So I have a very nice question. Did you try to, since it's an enteropathogenic microorganism, did you try or someone try to infect the Zebrafish by oral route? I mean, by adding the bacterium, I don't know, to the water, or just in the associated to the live food, something like this. I know that in your development stage, maybe it's not possible, but later, I don't know, just.
Yeah, so it is definitely possible. Some people have done it with E. coli strains, for example. You can do either gavage, oral gavage, it's possible. Yeah. You can also inject directly into the intestine from the outside. And you can also infect them with feed. I can say what I tried with Shigella, it didn't go very far in the sense that then I started to do different things, and this model works just too well to just, try to move to something else that doesn't work too well. I established 2 models where you can inject directly into the intestine, and the bacteria do persist for a period of time. But at the time we were doing a lot of survival assays, so I didn't have much interest on that model because you couldn't assess other things that I was doing at that time. But now with this research coming forward, it would be interesting to revisit that model. And another possibility was also at day 3 late, I used to infect paramecia, which are these little things that grow together with the fish in the water, and the people that work with zebrafish know what I'm talking about. They are little protozoa. You can infect them with Shigella, and the infection stays there for a while, and then you can feed infected paramecia to the zebrafish, and the infection will relocalize in the intestine in the natural way. I didn't continue very much on that because the association was very short, and the fish were not dying. But now with my interest switching more towards persistent infection, it will be interesting to actually have a look because that was not done. But it is possible to to infect them directly into the intestine, either with oral route, artificial, or actually natural via food,
Okay.
live feed even.
Thank you. In fact, I was asking you this question just because it's interesting to see in the case of the microbiota if there is any change or not in presence or absence of Shigella, just to see if Shigella can modulate or not the microbiota as it happens for other pathogens. So
yeah, absolutely. Yeah, I haven't done that consistently, but it's always been something very fascinating to me, and one day this will be done for sure. And in particular for Shigella sonnei, this is very relevant because people have shown that Shigella sonnei as an extra weapon also compared to other Shigella, which is the type 6 secretion system, which is important for bacterial competition. So perhaps it's even more relevant to actually look at how Shigella sonnei specifically interacts with the microbiome, and probably that gives them an edge compared to other Shigella.
thank you.
Thanks.
I was also wondering about the temperature, because we discussed the temperature limitation with David just before, and if I remember well, of course there are some link between the growth of the bacteria and also the metabolism that is temperature sensitive. And if I remember well, there was a question of persistent cells or dormant cells and things like that related to the metabolism. So how do you deal? Because don't you change the ratio between acute versus persistent infection also due to the temperature, the relatively low temperature?
Yes, it does happen. So these experiments I've shown here, they're all at 28.5 degrees, the standard temperature for the fish, and the persistent infection establishes very well there. But Shigella normally prefers 37 degrees, for example, because it's adapted to humans. You can still develop persistent infections at higher temperatures in the zebrafish model with Shigella. However, as you're saying, the ratio changes dramatically on the fish that will clear the infection and that will establish persistence, and while they will just die of the acute phase infection, because the fish become really, really heavily susceptible. Even when the temperature is raised to 33 degrees, then the initial bacterial load has to be very, very small, about 80 bacteria. So if you still want to establish a substantial number of persistent infections, otherwise the large majority of the fish will just die during the acute phase. They seem to be unable to control it otherwise.
So, and also with the resistance to antibiotic, do you think that you slow down by decreasing the temperature, the whole process that, or can you change it in terms of mutation and things like that?
Yeah, so these are some of the limitations, and that's why I'm actually working in parallel between the zebrafish model and also THV1 cells, which are human macrophage line, which where you can study some aspects, but not all of the persistent infections, such as antibiotic tolerance and AMR evolution. And we have to be careful with that, because it is true that at the same time, actually, paradoxically, what I see is an increase of the frequency of AMR, because I think the persistence stage is so dramatically changing that. But definitely with the change on the temperature, the frequency at which the mutation will occur will change. And people have shown that at 28 degrees versus 37, for example, the bacteria have different frequencies, of mutation. So it's a model, and it needs to be confirmed also with other systems, and that's what we try to do. So some aspects can be studied very well in the zebrafish, and other you need also to validate them in vitro with cell work and more and systems that are more representative of macrophages of humans, for example. Yeah.
Is there any mouse model?
So Shigella research has been quite handicapped by the lack of a real animal model for Shigella that was a mouse model, because the intestine of the mouse is resistant naturally to the invasion by Shigella. However, some people last year have published a mutant line where they basically knock out the inflammasome into the epithelial cells of mice, and these mice become susceptible to get infected with Shigella. However, still, it's not clear what happens to the macrophage side afterwards, and it's something I haven't explored. And the model is still quite limited. It was only from last year and only been shown with one serotype of Shigella flexneri. So it would be interesting to see how that progresses and if it's representative to study the multitude of different Shigella that are out there, and for example, Shigella sonnei as well.
Mm. And last question for the community more. Can you just give an idea of how many fish you inject to be able to make the videos that you show? How do you evaluate how representative it can be? How many larvae do you image for a long time up to the moment you will manage to get your concept visualized? This kind of. Can you discuss a bit about it?
Me?
Yes, of course you.
So it really changes. It really depends on what you need to show and if you have to do some quantitative data analysis. In terms of these graphs, for example, here I have literally, I think these are at least 50 fish to 80 fish per group, because actually the data can be spread. So you need to actually have many more representative things. But in terms of the videos, obviously, it's becoming more difficult to have large numbers. And so in some cases, I have things that have been observed in 5 fish 3 times. That's probably the minimum. However, now we're trying to quantify things also using a CD7, which is a sort of a auto imager, and have at least 24 to 48 fish for each of the different groups at each replicate, and try to analyze this in a more robust way when it's necessary.
But would you agree to say that in fact we observe a phenomenon in live imaging, and then we built an hypothesis that we verified with like genome editing, as you say, like abolishing neutrophil macrophages, treating with some drug to correlate the results with the first observations that we have?
Yeah, I totally agree that sometimes what you need is actually the hypothesis generation step. And I think that's what these videos are really instrumental for. So you watch that thing a few times, but you can build an hypothesis. And then these are relatively rare events that you are actually in the right position at the right time and everything right to observe it in a continuous way all the time. But actually, that's why then you move to other tests that you can do. You can scale up at different sample sizes to actually confirm that what your hypothesis was actually is in agreement with everything else. I totally agree with that. Yeah.
Okay.
So perhaps we can close, if we don't have any question from the audience, we can close the session. So on my side, I'm totally convinced now that the zebrafish larvae are really a powerful system that recapitulate many of the interaction of the physiological process and the immune process, and that has to be validated, of course, afterward with other systems. But it is a very promising system to model for in the free air perspective, because so thank you very much, because you show us a large panel of beautiful work and beautiful pictures, and how we can, from at the cellular level, but also more generally, address some very important questions regarding those pathogen interaction. So thank you, Christelle, David, and Vincenzo for your talk and your enthusiasm to present us during your presentation. Christelle, if you want to add something?
No, yes, I agree with you. I'm really happy to have this opportunity to hear about your work and to share my enthusiasm with this wonderful model with your different application. I saw that David was able to join us back. Yes?
Yes.
Okay. So yes, it was my pleasure, and I hope that also the participants get satisfied of the presentation. So if you have any questions that come afterward, do not hesitate to send us your question or discussion, and of course we will either transmit to the speaker or try to answer you. And thanks again to you guys for accepting.
Thank you very much.
so
It was
much.
really exciting presentation. Thank you very much.
Really, thank you a lot. Yes.
Have a nice day. Bye.